regarding your amino acids, your mostly from where we'll be getting the amino acids means we'll be getting questions mostly from the disease aspects, that is your amino acid disorders. That is one extreme of the questions, and another one aspect is from your normal chemistry, that is OH group containing, that is phenol group containing, what is simple, what is branched, what is amide group. So like that only we'll be getting a question. So this will be the two extremes of questions from amino acids, as especially from ionicity point of view. So in this thing you can see there are 20 amino acids and 21st is selenocysteine. Okay, so that is something you should be knowing it. And next, and this broad classification you can know or you needn't know, it doesn't matter much, but certain examples alone you should be knowing. For example, you should be knowing aromatic amino acids, that is phenylalanine, tyrosine, tryptophan. Okay, this is something you should be knowing. And heterocyclic also you should be knowing, that is histidine and tryptophan. So tryptophan is both heterocyclic and also an aromatic amino acids. And also your simple is glycine and alanine, that is the simplest amino acid. This is you should be knowing, and mostly question will not be happening. But your branched amino acids is there, especially your valine, leucine and isoleucine. Why this is important means from here only we will be having that propionic acid metabolism will be happening. So for that reason this is very important point. Next is your sulfur containing, you know. So how we are going to remember the sulfur containing means teen. In your teenage, remember your teenage and your sulfur. So cysteine for your sulfur. So this is very important and very confusing also. And OH you will be knowing, that is your T and S, that is your threonine and your serine. And amide group is amine will be asparagine, glutamine. So amide can also be asked as CONH2, and like this also they have asked the questions. So this classification is very important. So in this the focus points are, first start with your amino acids, 20 amino acids, your 21st amino acid is your selenocysteine, and then come to your simple amino acids, that is glycine and alanine, and then come to your OH group containing, that is your serine and threonine, and your especially your propionic acid, that is got from your valine and leucine, and amide group is glutamine, asparagine. That's also, this will be introduction for your amino acid chemistry, and especially your benzene ring is concerned. So with the benzene, phenyl, that's all. Phenol is thyro, O is there, so tyrosine, phenol. Imidazole, histidine, very important. Okay, proline, pyrrolidine, and indole is also O is there, so tryptophan. So you shouldn't confuse between phenol and indole. Phenol is your tyrosine, indole is your tryptophan. So this is very important. So how we are going to clear this thing means indole. So N, it should end with IN, that is a tryptophan. But phenol, phe, so it should end with tyrosine, E, that's all. So this is something you should be, you should not mess it up. So tyrosine, phenol, indole group, tryptophan. Pyrrole, proline you can remember, imidazole, histidine, that's all. It has a 2I, it has a 2I, that's all. So next, coming to your polarity. So this is not that much important because it will get very much confusing here. But one thing you can have is your aspartic acid, glutamine. So both has an acid name in it, so that will be your acidic. And basic is what? Asparagine, lysine, histidine. So this is very important here. So next, coming to your metabolic fate. So metabolic fate is according to your glucogenic and ketogenic and both. So how we are going to remember this means, just remember ketogenic, that is a leucine. Definitely one of your options will be leucine, so ignore that option. And both is what? Both is your pitfall is the mnemonic. So what is the pitfall means? Phenylalanine, isoleucine, tyrosine, tryptophan, lysine, that's all. So pit, PITT, fall, that's all. So fall is for your both fat and your glucogenic. So there is pitfall mnemonic is important, and all the others is your purely glucogenic. So totally 20, 14 are glucogenic, leucine you should be remembering, pitfall you should be remembering. Next, nutritional requirement based classification. So in this thing, essential. So essential is what you should be remembering here. So tryptophan, valine, leucine, isoleucine, lysine, phenylalanine, methionine. So these and all you should be taken in your diet, and it's not MCQ point of view, it's not that much important, but your semi-essential arginine and your histidine, that comes under semi-essential. That keep that alone at least. So next on that note, going to your urea cycle disorders. So urea cycle, okay. So first, according to the site, both your cytoplasm and your mitochondria, and the most important step in your urea cycle is, there is two things you should be remembering. One is your ornithine transcarbamylase, ornithine carbamoyl transferase, okay, and then there is an alpha ketoglutarate transamination. For transamination, what is the coenzyme we should be requiring? That is a vitamin B6 is required there. And then first in your blood this is happening, and then it is going to the liver, and in your liver only glutamate dehydrogenase is coming into play. So first alpha ketoglutarate, that reaction will be happening. Then glutamate is like a transporter, and that will transport this amine group structures into your liver, and in your liver your glutamate is adding. So glutamate, it becomes glutamate dehydrogenase, and from there only we will be getting what? Your urea formation will be happening. For urea formation, what is the rate limiting enzyme? That is your CPS1, and we require what? NAG, that's all. So these details alone is more than essential, okay. So this is a question that you should be knowing, okay. So this next, urea cycle disorders. Don't go into much details of the urea cycle disorder. You should be just knowing this. Start with your alpha glutarate, go with your glutamate, and then go into your cell. In your cell it is glutamate dehydrogenase, ornithine transcarbamylase, and ornithine transferase, that you should be knowing. And then CPS1 is your rate limiting enzyme, that's all you need to know. And there is an ornithine transporter. The ornithine transporter doesn't transmit ornithine, it transmits only citrulline, okay. First option is your citrulline, next option is your ornithine, if at all they are asking a question on that, okay. So this is very very important. So next, coming to your urea cycle disorders. So especially in your urea cycle disorders, first CPS1 is problem means hyperammonemia type 1, okay. Ornithine transporter. So ornithine transporter, many names are there. So hyperammonemia, yes, ammonia can get increased. Hyperornithinemia, homocitrullinemia, okay. Carbamyl phosphate plus lysine. Next, whenever your ornithine enzyme, it's a transporter, here it's an enzyme. So enzyme means that is your type 2. 1 means that is your CPS1, means that is your hyperammonemia type 1. And see this synthetase, okay, from arginosuccinate synthetase is not getting formed, means your citrulline is getting increased. So this is called a citrullinemia. When your arginosuccinate is not able to break down, means we are going to have arginosuccinaturia. Arginase, that is the last enzyme, is your arginase. So arginine will be increased, that is hyperarginemia. So all these will be your this thing, and presentation will be more or less will be the same. Basically your crying child, abdominal pain, vomiting, aversion to protein food, this is very very typical, okay. Aversion to protein food, and your growth retardation, mental retardation and all point towards your amino acid disorders, that is more precisely in your urea, because urea is an irritant of your brain, okay. So this is very very important. I am just telling it again. CPS1 hyperammonemia type 1, your ornithine transcarbamylase, ornithine carbamoyl transferase, T4T, T4T, T42, hyperammonemia type 2. Ornithine transporter, so transporter means it has many other names, that is your homocitrullinemia, hyperornithinemia, hyperammonemia. Your arginosuccinate, synthetase, lyase, arginase, this and all, it's simple, you will be knowing, and clinical presentation you should be knowing very well. So next, coming to your phenylalanine. So phenylalanine is the first, it is first essential, yeah, we know it is not synthesized in our body, that we know, okay. So what is the point you should be knowing means the breakdown products, that is your phenylalanine acetate, phenyl lactate, phenyl acetate, or whenever acetate is adding, will be having a peculiar odor to it. What is the odor here? Mousy odor or musty odor. So what is the screening test you will be using? FeCl3, ferric chloride, 10% ferric chloride. So on adding ferric chloride, you will be getting what? Your leafy green precipitate is changed, color change is present, means that is possible a case of your phenylketonuria. And how are you going to confirmation? Confirmation is by Guthrie. So screening has a E. So screening has a E, that is your FeCl3, and then confirmation C looks like G, so that is your Guthrie test. So this is very very important point. So your test tube, whenever your test tube is shown and green color is shown, means that is your FeCl3 screening test. But whenever your this thing, a culture or this thing, a colony is shown here means that is your Guthrie test. So this is, you shouldn't be missing it up. So what is the organism we are using? First we are taking a plate and then we are spraying Bacillus subtilis, and then to that we are adding what? We are not adding phenylalanine. If phenylalanine is present in the culture, the bacteria is going to utilize that and grow. So if phenylalanine is present, the colonies will develop. Phenylalanine is not present, it will not grow. So this is the inference you are going to make from your Guthrie test. So what is the final agenda of this thing is treatment. So treatment is, first it is an essential. So we have to restrict what? We shouldn't restrict phenylalanine per se, or we should supplement with tyrosine supplementation. Okay, it is required in phenylketonuria and it is started less than 10 days after birth. This is very important point. And till long, how long you have to give? Till lifelong. Okay, so this is very important point. So phenylalanine, the treatment aspect is important for AIMS. Tyrosine supplementation also given. First stop your phenylalanine, and then all this should be done within 10 days. That's all, till lifelong. So next, coming to your tyrosine. Okay, so tyrosinase, that is a copper containing enzyme that you should be knowing it. And what is the compound we need? Dopa, first thing, and next is your melanin. First comes dopa, next comes melanin. This alone you should be knowing. And then in albinism, what happens? So in albinism, the melanin is not formed. There is a mutation in your tyrosinase. That's also, there is a uniform absence. This is the key word here. And whenever in your copper deficiency, there will be melanin, all the M will be decreased. That is, your malabsorption will be there, malnutrition will be there, melanin, melanin will be there. So this is for your tyrosine. So one more byproduct of your tyrosine is catecholamine. So one is melanin, another one is catecholamine. So what is the enzyme for catecholamine? Means that is tyrosine hydroxylase. Okay, so tyrosine hydroxylase and your dopa decarboxylase, dopamine hydroxylase. So like that it will be going. But here it is only tyrosinase. That alone you should be knowing here. So here basically you should be knowing epinephrine and norepinephrine. See, first we get what? Dopamine, next norepinephrine, next epinephrine. You don't need to know much details about this thing. And this direction is important. S-adenosyl homocysteine to S-adenosyl methionine. So this is very important. Why? Because it is going to act as a methyl donor. So what is the methyl donor? It's your S-adenosyl methionine. So catalyzed to form metanephrine's vanillyl mandelic acid, urine. So this is very important. So because of this only, we are going to, in pheochromocytoma, we are going to measure vanillyl mandelic acid in your urine. You are not going to measure your epinephrine or norepinephrine, because that is anyway is going to get catalyzed and you are forming vanillyl mandelic acid. And this excessive formation of metanephrines will be seen in your pheochromocytoma. This aspect is very important. That's all. This aspect is super important. And next, coming to your tyrosine. Yeah. So tyrosine anabolism we've seen. So from the tyrosine only we are getting melanin, and from the tyrosine only we are getting catecholamine. So two most important compounds synthesized from your tyrosine. So next is your this thing, catabolism. So what is the catabolism for your tyrosine? So catabolic disorders, that will be having tyrosine transaminase, TT. 2T is there, so it has to be tyrosinemia type 2. Hydroxylase, it doesn't have any T, so it has to be 3. So this is how you should be remembering. So in your type 2, that is your tyrosine transaminase problem, that is your tyrosine will be accumulated. But in your parahydroxyphenylpyruvic acid, that is PHPP, like that only they will be giving in the question. It is PHPP. So whenever you are having a PHPP, that is your tyrosinemia type 2. And then ultimately we are going to have this homogentasic acid, and this is going to precipitate as alkapton bodies. You know where it is getting deposited. And this in alkaptonuria will be hitting homogenticate oxidase. That's all you should be knowing. So in alkaptonuria, homogenticate oxidase. Tyrosinemia type 3, that is your hydroxylase. Type 3 will not have a T. That's all. So it's just a tyrosine hydroxylase or decarboxylase, and in this your PHPP will be accumulated. So why this is important means, because the substrate also doesn't have a tyrosine in the name, the product also doesn't have a tyrosine in the name, the enzyme also doesn't have a tyrosine in the name. So it is very difficult to guess. But your tyrosinemia type 3, that is very important here, because homogentasic acid, by this time you will be knowing alkaptonuria, and your tyrosinemia, tyrosine transaminase, that is kind of explanatory only. So this is very important. So what we have seen tyrosinemia type 2, we have seen tyrosinemia type 1. What is tyrosinemia type 1 then? One is called as what? Tyrosinosis. Here the fumarate acetoacetic acid, that is getting increased. So whenever that is getting increased, we are going to have rancid butter smelling urine or boiled cabbage. So this is important. So first is your boiled cabbage. That's all. Boil your cabbage. So this is the main motto behind this thing. So type 1 is our boiled cabbage, and type 2 is our tyrosine transaminase. It has a 2T, and type 3 doesn't have a T, that is a hydroxylase or a decarboxylase. That's all you should be knowing. So tyrosine done. Next, coming to your tryptophan metabolism. So in tryptophan metabolism, I already told you we need what? Vitamin B6 for your niacin formation. We need vitamin B6 for synthesizing B3. This is the point, it is important. So tryptophan B6 required to form B3. So serotonin is there, melatonin is there. So don't confuse between melanin and melatonin. Melanin from your tyrosine, melatonin from your tryptophan. That's also here one pathway, that is kynurenine pathway, very important for AIMS. So here, what actually happens here means, what is the test you are going to do? So we are going to do a check for your tryptophan load or stress test. So whenever tryptophan load is given, xanthurine in urine is increased. So whenever in case of your B6 deficiency, this will the load will not be increased. So this is done to find out latent B6 deficiency. So this latent word is important. Okay. So next, coming to your so whenever your xanthuremic acid levels are increased, it suggests B6 deficiency. So this is very you should be doing. And also in your pellagra, that is your B3 deficiency also, it can be due to B6 deficiency. So this is very important. Okay. So next, coming to your that's all you need to know. You shouldn't need any other thing to confuse things. Tryptophan, B6 remember, B3, serotonin remember, melatonin remember, kynurenine pathway remember. We are going to check our primary concern, though it is kynurenine pathway, our primary concern is to confirm your B6, because without B6 we are not going to get B3. So this is the most important aspect of your tryptophan metabolism. Okay, so that alone we are getting here. Next, coming to your serotonin and melatonin. So serotonin and melatonin, what you need here? So 5-hydroxytryptamine, that is a serotonin, is used as a neurotransmitter in your GIT cell, and it also helps in motility here. So carcinoid tumors, it is called as what? Argentaphenomas. So after degradation, it forms 5-hydroxy-indolacetic acid. So this is overproduced in our carcinoid syndrome. Don't confuse between pheochromocytoma and carcinoid syndrome. In pheochromocytoma, that is your your vanillyl mandelic acid, that is tyrosine metabolism. So pheochromocytoma is tyrosine metabolism. Your VMA is getting increased. But in your carcinoid syndrome, your 5-HIA, that is getting increased. And your melatonin, what is the main function of your melatonin is pineal gland hormone. That is a diurnal variation. So this is very important. So next, coming to your heart nerve disease. So how you can remember means, there is a H, there is a T, add your 5 to it. That is a 5-HT serotonin. So heart nerve disease is concerned with your tryptophan. So what is the main problem here means, it's a neutral amino acid transporter defect. That's all. No enzyme problem in your heart nerve disease, it's a transporter defect. Because of this, there will be pellagra like features, not pellagra. So it is your dry skin, dementia, dermatitis and diarrhea. It will be similar to your pellagra, but it is nothing to do with your B6. So go for your heart nerve disease. Nothing. It's an amino acid transporter defect, especially in your present, where it is present in your GIT mucosa and your kidney tubules. That's all. It's not present in your liver. And this was a question they have asked once. So this is very important. And what is the urine we are going to have? Urine will be having what? Blue color urine, blue discoloration. So our tryptophan is done now. So next, coming to your methionine. So methionine and cysteine are your sulfur containing amino acids. So it is an essential that we know. So what is the main focus here? It is your homocystinemia. So it is methionine, but the disorder is what? It is not methionemia or like that. It is homocystinemia, homocystinemia type 1, homocystinemia type 2. So basically, homocystinuria, we know that subluxation of your lens and the difference between your morphin and this thing, that we know, knowing that alone is important here. No other point is that much important. That's all. So yeah, whenever there is a cystothionein beta synthase, and this is important. Why? Because your pyridoxal phosphate, which means B6. So in your type 1, there is a vitamin B6 pyridoxal phosphate is needed. In your type 2, that is S-adenosyl methionine, that thing will be coming into play. That methyl donor, that will not be happening. So that is your type 2. So this is very important, and most of your amino acid metabolism takes place in the liver cytosol only. Okay, and except your urea cycle disorder, that happens both in your cytoplasm and also in your mitochondria. So next, coming to your histidine. So especially in your histidine, it will be similar to your fructose metabolism or galactose metabolism. Okay, so here you can see histidase deficiency benign. But your urocanase, that is a urocanic aciduria, they are yet to ask a question on urocanic aciduria, and it has to do with your histidine. Okay, so this is very very important. And what is the test you are doing in your histidine? That is a figlu. So figlu excretion in urine denotes histidine. That's all. And then to detect latent folic acid deficiency, we are giving what? Histidine load test or histidine stress test. So this is called as figlu excretion test here. So your tryptophan load test is done for your B6 deficiency, that is latent. Okay, but in your latent folic acid deficiency, you should be doing histidine load test. So this point is very very important here. Okay, and then coming to branched chain amino acid catabolism. So in your branched chain, three things are important, that is your valine, isoleucine, leucine. So in this the most important is leucine, and also propionic acid is got from here, that you should be knowing. You can see this propionyl CoA especially from your isoleucine. And what is the thing you should be remembering here? Your branched chain, burnt sugar, especially your valine. Okay, so valine, maple syrup, urine disease, that's all you should be knowing here. So valine and isoleucine, propionyl CoA is going to get, and this is a glucogenic. And your isoleucine, it is both ketogenic also. And then leucine has isovaleric CoA, so isovaleric aciduria, cheesy order in urine. So what is the deficiency here? Isovaleryl CoA dehydrogenase, that's all. So the questions from branched chain amino acids are pretty small, but it is pretty confusing. Why? Because three lines you should be knowing. First, examples, that is your valine is there, isoleucine is there, leucine is there, V is there, I is there, L is there. Remember, V is for your glucogenic extreme, L is for your ketogenic extreme, and I is for both G and K. This is how you should be reading. Okay, and your L disorder is, L has nothing to do with your, your isoleucine has nothing to do with isovaleryl CoA. So your leucine has to do with isovaleric CoA disorder. Your isoleucine, no specific disorder is there. And your branched chain, that is your valine for your maple syrup urine disease, MSUD. So here we are going to have burnt sugar, and a cheesy order is isovaleryl. That's all. So there is no disorder for your isoleucine, or maybe not, they are not tested in our setup. That's all you should be knowing. Okay, so this is for your whole amino acid disorders. You got it? Yeah.